So hello, everybody. My name is Leanne Reynolds. I am the Director of Education at the Squam Lakes Association. I almost said that I work for the Science Center. That is not true. But this Science Pub series is sponsored by the Squam Lakes Natural Science Center and the Squam Lakes Conservation Society, as well as the SLA. So it is a collaborative effort. And just a few announcements. We have two more science pubs in the series. So April 13th, we'll be exploring our nocturnal neighbors, bats of New Hampshire. And then on May 11th, we'll be learning about zooplankton. And so, but tonight you're here to hear Natalie. And I am gonna give a quick little intro to Natalie before I turn it over to her. So Natalie grew up in southern New Hampshire, and she has a Bachelor of Science from Cornell University and a PhD focused on moss ecology from the University of Alberta. For the past 20 plus years, she has been employed in field research through Cornell University, and her primary research site is Hubbard Brook Experimental Forest in Woodstock, New Hampshire. Together with coordinating and leading the long-term tree surveys, she has personal interest in factors that influence the occurrence and diversity of tree epiphytes, survival of tree seedlings, and the demography of round-leafed orchids in the forest understory. So I will go ahead and turn it over to Natalie.
Hi, everybody. Thanks for coming. Just a couple of disclaimers. As you heard in my intro, I am not a lichenologist. I'm an admirer of lichens. So I hope you will also become an admirer of lichens. It's easy to do. And also none of my studies were specifically done at Squam. But I've been in New Hampshire a long time, and I was once a bryologist, a person who studies mosses, liverworts, and hornworts, although there's really no funding for those type of people. So I have had to become another person. But without further ado, it's my real honor to speak to you tonight on behalf of the epiphytes that live around us, which include the lichens, the mosses, and the liverwort. So what are epiphytes? Well, epi means upon, and phyte is a Greek word for plant. And so we could think of this as a story about once upon a plant. There lived some very interesting organisms. And a lot of people think epiphytes hurt their hosts, what they're living on, and that is untrue. And in fact, if you move slowly enough, like the person in the lower corner of this slide, you might get some growing on you. And they're really just using you as a leg up in the world to get a little higher, a little closer to the light, a little closer to the cloud base, things of that nature. They do not in any way hurt the hosts that they're growing on. So once upon a plant is the story, how we're going to start our story tonight. And we'd like to learn a little bit about our main characters. So our main characters are in two very different kingdoms. We have the lichens that are fungi. Actually, they're much more than fungi. They're very complex, but I'll get to that in a sec. And we have the bryophytes that are plants. And so these two very different kingdoms these organisms have come to have very similar ecology. But first, I'm going to go through some of the differences. So the way that I've seen lichens defined that I like the best is lichens are fungi that have learned to farm. And what they're farming include algae, cyanobacteria, and now we even learned that it also may include yeast, so other fungi. And so the fungi, who are the main organisms named when we name a lichen, create homes for these other organisms and live in what we call a symbiosis. It's a mutually beneficial relationship where the algae and cyanobacteria are photosynthesizing, and the lichen is providing a constantly protected environment. And so they'll slow the drying, and they make the temperature more moderated, things of this nature. But then the bryophytes are actually plants. So they photosynthesize by themselves, and they're not as complex as the lichens. They can live on their own. They're not a whole host of organisms living together. And so if we go back to the lichens, the two main ways that we superficially divide the lichens would be the crusts. So these guys over here, where the actual body of the lichen is embedded in the substrate and you don't really see it. There's no like real 3D nature to it. And the macro lichens, which as you can see from this photo, have a lot of dimensionality to them. They're very 3D, they're bigger, macro. And these are the lichens that I'll be addressing in my talk tonight, even though the crusts are also important. And then the bryophytes include hornworts, which I'm not going to talk about. I'm going to talk about liverworts. And so these tend to be very flat. And another important difference from the mosses is that their spore-producing structures, which you see here, they are very short-lived. And so think of them as fairly flat, and you rarely see their sporophytes because they just don't last very long. Whereas with the mosses, they're the way they create their leaves from the stem, they're very spirally oriented. So they tend to be much more three-dimensional. And they have long-lived spore-producing structures. So you're often going to see them with their spore-producing structures. Now we're gonna test you. We're gonna see if you can play the lichen versus bryophyte game. So these are two, I wish this could be more interactive. I wish everything about life could be a little more interactive than it has been for quite a while. But if you'll bear with me and in your mind, you could think of the answer. So I'm gonna put the cursor, I'm gonna circle, and then I'll tell you the answer. So this organism right here, and you can see these cups here are how this organism produces its spores, and this is a lichen. This white patch right here is also a lichen. It's one of the crustose lichens, whereas this guy is a macro lichen. This fellow down here, I can hear you all answering me. It's a bryophyte, it's a bryophyte. This is a moss. And also there's a second species of moss here, which is much more of a cushion shape. We go to the next picture. We can see here, this fellow is kind of light yellowish green. This is a macro lichen. And then there's kind of a reddish, very flat, that's your key, very flat to the substrate. That is a liverwort, so that's a bryophyte. And up here we have two species of moss. So moss are bryophytes, plants. I could go into genera, and I'd love to do that, but you'd all be bored to tears. So we're just going to try and get you to recognize a lichen, a fungi from a plant would be my goal for you tonight. And I do have another challenge for you at the end, but let's try and just get you thinking when you see the pictures, is that a lichen or is that a bryophyte? One more test for you. Over here, this was taken in my yard. So here's a macro lichen number 1, And you can see how the color changes. Macrolichen number 2, moss, 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 liverwort. Aren't they cute? They're so flat, but they are kind of cute. And then we've got another macrolichen down here, and then you can see a bunch of moss. And don't forget, there is crusts. So if you ever thought these were like some kind of disease or something undesirable, these are perfectly desirable and wonderful creatures. I think a lot of people maybe misinterpret crusts, but crusts also do not harm the host. So I'm going to spend most of the talk talking about the actual similarities rather than the differences. So now that's all I'm going to say about the differences. All of these organisms that are epiphytes are desiccation tolerant. And this is really different from being drought tolerant. So if you think about a cactus, that's not what we're talking about. So a cactus is really good at living in a dry place and keeping water in its body. But these guys don't try and keep water in their body. They kind of go with the flow. So when it's wet, they're wet. When it's dry, they dry out. They really dry out. Almost no water content. This is what we call desiccation tolerant. These are the kind of organisms that you can send to the moon and back. It's pretty neat. And it also has some bearing on how they handle their water ecology. And it's really analogous to animals. So we talk about ectotherms and endotherms. We're endotherms. We regulate our temperature inside our bodies. But a butterfly, for instance, is an ectotherm. It's going to get cold when it's cold and warm when it's warmed up by the sun. And in the same way, we have plants, and in this case, fungi, who can regulate their water outside of their body. So they're called ectohydric. Ecto, outside, hydric for water. And whereas most of the plants, like in my house here, the house plants in the background, and most of the plants that you see when you go outside, they're endohydric. They're actually maintaining water pressure within the plant body. And it's really different evolutionary trajectories in the same ways that animals have these really different evolutionary trajectories. So that's my little lecture. I'll try not to lecture you too much, but we have to get you to understand how different and unique their water relations are, because it has a bearing on everything else I'm going to talk about. And also has a bearing on really appreciating what these organisms are telling you about the environment when you find them. One of the consequences of trying to regulate their water content outside of their body is that they have a very large surface area to volume ratio. So they tend to have lots of little nooks and crannies where they can trap water. As a consequence of this, they're also getting all their nutrients from outside the body. They don't have roots. They're not drawing anything up from the tree. They may get things that come down the trunk of the tree in the rainfall and the stem flow, but most of what they're getting is from the air. And this makes them really sensitive to air pollution. They're also, as I mentioned, very three-dimensional again for the water holding capacity, but it makes them basically little jungles. They're little forests within the forest. And so they're important habitat for things that we don't think about very often, like rotifers and tardigrades and collembolans and little spiders, little beetles, all kinds of things. They're habitat, and then they're also sometimes food for animals, and we'll actually talk about that a little bit. And because of their really kind of special relationship with water and moisture can be very easily disrupted. If you cut a tree down and you create an opening, or there's a forest blowdown, there's a natural disturbance, and you create a big opening, you really change the moisture dynamics of that site. And so they tell us a lot about the continuity of a forest on the landscape. And they don't necessarily, I would argue, they don't tell us the age of the entire forest. I don't think they necessarily tell us if a forest is old growth in the most pure sense, but they tell us that this site has been continuously having trees on it and continuously having microclimates which could sustain these species. So I'll be talking about that a little bit too. This is kind of like the outline of my talk. So again, this is a story of the epiphytes. I really don't take any credit. Here it is just it's a process of trying to pay attention to what they're trying to tell us. And they speak of water. They speak so loudly of water. They tell us where the cloud base is on the mountains. And in particular, this lichen here, called the ghost antler lichen, this guy, Pseudovernia cladonia, is one of the lichens that you can use pretty accurately to go on a mountain and know how often is that mountain and where on that mountain the cloud base sits for about 50% of the time. So it's pretty neat. They're really tied in to water and not thinking about water as a stream or a lake. We're thinking about micro water. We're thinking about drops of water, fog, dew, things like this. This is the water that they tell us. So just to make sure, I know you're all going to get this because this is a third grade science activity that I designed last year I was hoping to do it with my daughter's science class, but because of COVID, I could only do it with my daughter at home. But we went through 10 reps. We used 3 growth forms. So on the left, you can see, we'll call this the hanging. And we got in the middle are the big old ruffles. And then on the right here are the flat guys, which are called folios. We call them flat. These guys are actually folios too, but they're much more 3-dimensional. So we distinguish them as ruffles. And so what we did to look at how form and water holding capacity are related to one another. Pretty simple. All you need is a scale and a spray bottle and some enthusiasm and some lichens. So you weigh your lichen dry, and then you take it over and you spray it with water till it's fully hydrated. So we decided this was about 20 sprays. And then we shook it. So shake the extra water off. And then you put it back on the scale and get your second weight. And then by this very simple formula here, you can get to the percent of dry weight, the percent of the dry weight that water can, that lichen can hold in water weight. So what did we find with our 3 forms? How is lichen form related to water holding capacity? So the hanging, you can see, was an average of about 5 times its dry weight it could hold in water. Pretty good. And then the lowest one was the folios, the flat folios, which was still more than 2.5 times its weight in water. Pretty nice. So that they're holding all this on the outside, right? But if you think about it, this really slows down. It controls how fast or slow they're going to dry out. So they can actually hold this water there and dry out more slowly. And for them, being wet is being awake. Being wet is being alive and able to gain carbon and grow and reproduce. All this requires wetness. Being dry means being asleep, waiting. Time passes. Being wet is being awake. So as I mentioned, this three-dimensionality, this being open to the air, getting everything from the air and the rainwater, the fog, being a very large surface area to volume ratio means you're pretty thin, very thin, and you've got a lot of surface area to collect on. It makes them very sensitive to pollution. Some of them are sensitive in the sense that they die. And so this guy here, pseudocephalaria, this is a photo I took when I was working in Acadia. This lichen doesn't exist here anymore. This is a very sensitive, pollution sensitive lichen, and we've lost many of those from the state. But this lichen here, down the bottom, is sensitive to pollution, but it's an indicator of pollution. This thing loves nitrogen. And I've noticed in places where the dogs pee a lot, you can usually find this lichen. So it's an indicator of nitrogen levels. So the epiphytes are really good indicators of many things, of moisture, of pollution, and we'll go on to this list. But because they're out there, they're just out there absorbing everything and not controlling anything within themselves. They're just taking what lands on them and working with it. So this is a study I was involved in. I started with my work in Acadia, sort of in the mid 2000s, and then in 2011, to 2013, we added some of these. They're called class 1 areas. They're areas where they're supposed to be making sure that the air quality is still high. So we have the Great Gulf, the Dry River. These are wilderness areas in the White Mountains. We have Lye Brook here in Vermont. And this map is showing, the darker it is, the more nitrogen deposition has accumulated over time. And so one of the things you'll notice is there's a pattern of decreasing deposition as you go east. And that's because most of the pollution sources are in the Midwest, except you can see here also the urban center around Boston, you can see is much darker. And the other thing you'll notice is that you can see the Green Mountains here, and you can see the White Mountains here. And so places of higher elevation get higher deposition. And unfortunately, places of higher elevation are also usually where there's really good lichens. So, and all of our sampling sites here, except for Acadia, were higher elevation sites in the mountains. So what did we find? Well, first of all, how did we find it? So one of the things we did was we thought, well, how can we we can't just use the diversity because we know so many of the sensitive species just aren't around anymore. So we need something that's still going to be there to tell us how good or bad the situation is for the lichens. So we developed these thallus condition scores. So you can see here, the best is the number 3. This lichen is healthy, happy. It's so happy that it's able to reproduce with spores. Number 2, it's pretty happy, but it's not reproducing with spores. It's only doing asexual reproduction. Number one, it's got some asexual reproduction still, but it's also got some visual damage to the thallus. And then the poorest, it's not reproducing at all, and there's a lot of damage to the thallus. So what did we find? Well, first of all, if you look at these graphs, it's just showing the nitrogen content. So the more nitrogen that falls on you, the more nitrogen you're going to contain. So remember, they reflect their environment. And you can see here, it's a little bit backwards. So because of the alphabetical order, but if we think of it backwards, this is west in Lye Brook in Vermont, and we're going east as we go to the left. A little counterintuitive, but because the alphabetical is against the compass here. But you can see that Vermont's the worst in terms of having higher sulfur and nitrogen in the lichens, and the best was in Acadia, which is what we would expect. And that really closely corresponded to the condition of the lichens. So this is our thallus score. Remember, 0 is the really awful. The thallus is like 50% damage. There's no reproduction. And up at 3 is where we have the happiest lichens. So you can see there's none of the happiest lichens at Great Gulf or Lye Brook. We only start getting really happy lichens. Dry River, we found a few, and then at Acadia, we actually had the most. And you can see the other thing you want to see is that the shape of these. So at Acadia, most of the lichens are pretty good. They're pretty good. They're in the 2 range. Whereas in these sites where there's more pollution, like at Lye Brook, most of the lichens are in the 1, which, as you recall, they have some damage and there's no sexual reproduction. So we're starting to lose sexual reproduction pretty quick. And this was present day. This was 2011 to 2013. So although the Clean Air Act has had great impact on improving air quality, the lichen response is very slow. And part of that might be because it's a cumulative effect on the lichens. So although there's less coming today, all the stuff that came in the past is still kind of impacting them and impacting the bark chemistry of the trees. Okay, moving right along. So obviously the epiphytes also speak of the trees that they're growing on, and some people will ask, well, are they species specific? Are there some that only grow on certain species of trees? And they really don't see the world that way. To them, it's just all a continuum. And they have a special part of that continuum that they can grow on. And it's not necessarily related to the species of the tree, particularly when pollution has imposed itself on the natural gradient. So one of the other studies I did was in Acadia National Park, looking at the effects of the acidic fogs that they get there. The acid fogs come up the eastern seaboard and make landfall at Acadia. And they had measured some fogs that were of a pH as low as 2.7. They were black. They were black when they put them in the funnel. So pretty serious acid fogs in Acadia. So I was interested to see if the tops of the trees would be more acidic than the bottoms of the trees. We know that in places where there's little pollution, that the pattern is that the pH actually goes up as you go up the tree, because the bark is younger and it has more nutrients in it. But I did find the reverse at Acadia. The lowest pHs are at the base of the tree. The highest pHs are at the base of the tree, and it gets more acidic as you go up the tree. So pollution basically reverses the vertical gradient of pH on trees. And I would guess that if this is the case in Acadia, that it's probably the case here. So I was talking earlier about how the epiphytes don't see species specific for trees. They just see this continuum. And so you can see here that, that there's some maple and spruce, these are red maple and red spruce, that would have the same chemistry. So a lichen isn't going to care whether it's a maple or a spruce. They're just going to care about the chemistry. And so that surprises people, but I thought I'd let you have it. What else do epiphytes speak of? They speak of sort of the interconnectedness of these little worlds that they create. And so, as I mentioned, there's rotifers that live particularly in the mosses. This is a tardigrade. These are not my photos. These are not my photos. These two are my own. On this slide, most of these have been my photos, but I don't want you to think I took these. But anyway, these tardigrades is on a moss. This is really high magnification here. But tardigrades, clemelins, they live in these miniature jungles. And then these bigger guys, I think these are the like dinosaurs of the epiphyte world. The slugs and the caterpillars and the snails, they're eating the lichens and the mosses. Although this guy, I think, is just dispersing. You can see he's got a bunch of Ceridia on his fur. So I think he's just kind of dispersing the lichens. And then as you can see with this hummingbird nest, that many birds use mosses and lichens to build their nests. Camouflages them. Interconnectedness is generally thought of as having a positive connotation, but it can be negative when you're at the bottom of the food chain. And it can be really negative if a new herbivore comes into the system. So I should have pointed out this fellow here, which you may have seen in your garden or some of his friends, is Arion species from Europe. So these are not native to North America. And they eat a lot, and they do eat lichens. And so one of the questions I came up with at Hubbard Brook was, could these invasive slugs be part of the problem with some of the lichens that I saw as being damaged and seem to be decreasing in their numbers? So we put together a study to sort of look at the herbivory on the lichens at Hubbard Brook. And we were looking at snails. These are native snails. We're looking at snails and slug herbivory. This is what snail herbivory looks like. They make these little trails in the thallus. They generally eat the upper part of the thallus, which has the algae in it. And we found that if you're going to look on the landscape and think about where this grazing impact is the worst, there's a lot of significant relationships. It's worst where east facing, lowest elevations, where there's higher soil pH, where the plots are flatter, and where there's more hardwoods. And unfortunately, a lot of these landscape characteristics are also where a lot of the most vulnerable lichen species tend to be. They're the richest sites that stay moistest the longest. And so there were differences as well in how much grazing we saw on the lichens. From this is an example of a almost never grazed lichen here, Amshagia, to an always grazed lichen, Lobaria, also called the lungwort. Sorry, I need to drink water. It's a good time to take a pause because lichens and mosses on trees also speak of time, and I spoke about this earlier. They're really good indicators of the continuity of forest in a place. And I'm lucky enough to have a place like this be my backyard. I've been finding a lot of what are called old growth indicators in my backyard, which I know is not old growth, but I also know it was never cleared for pasture. And I think we need to pay attention to these places on the landscape. We have so little old growth. We need to also value these sites where the forest has truly been continuous. There's been maybe some firewood taken. Maybe there was sugar bush done there, but there's been constant trees, and that really makes a difference to a lot of the creatures, including the epiphytes. So one of the ways you can see this quantitatively is that, so here on the bottom, we've got all the species of trees. So this is beech, sugar maple, striped maple, red maple, yellow birch, white ash, and cedar. And you can see that no matter which species it is, you'll find these cyanolichens always more likely to be on the larger trees. And part of the reason for that is that it takes time for them to establish. It takes time for them to grow. And this time is only there if the tree's been there long enough. It may also be because the bark characters that they need in order to establish take time to develop. But they tell us a lot about time. And so I just wanted to close with a challenge. It's always good to have a challenge in your life, especially now when everyone's looking for something to do. Well, the epiphytes need you. They need you to look for them. They need you to document them. And they need you to think about why they're there. And so I've selected 3 that I think are really good indicators. They're really easy to identify. They'd be hard to confuse with anything else, although I'm happy to verify IDs. And so I'm going to introduce 3 challenges. First is your lichen challenge. So Lobaria, you've heard me talk about this lichen a bit. It's also called the lungwort. People, it's good. I mean, I think in the past is because they thought it looked like the alveoli of the lung, but it's also a good name because they're fairly sensitive to air pollution. So where they occur, your lungs are also happier. But be aware, these desiccation tolerant creatures tend to look very different when they're dry and when they're wet. And so when they're dry, they tend to be crumpled in, they're smaller, they're lighter colored usually because they want to reflect off the heat. And they're just usually harder to notice. But when they're wet, They're typically greener, they're more three-dimensional, they're more spread out. And so actually one of the most fun things to do if you're really bored on a dry day is to go out with a spray bottle and find yourself an epiphyte and wake it up. So it's really fun because people think, you know, animals are the only thing that move, but boy, I tell you, take your spray bottle out there and you're going to get a surprise. These guys can move when they get wet. They all open up. They're just, it's just, I never get tired of seeing it, to tell you the truth. I love it. You'll love it too. Please try it. So your first challenge, Lobaria. The only thing that looks like this out there. It's kind of looks like a big leafy thing on the tree. Picture's worth 1,000 words, but I'm going to give you some references at the end if you want more text on this. And this guy's in my backyard. This guy is up in the Quincy Bog Pasture Trail in Rumney. This guy here was in the Hemingway State Forest in Wanalancet, Tamworth. I'm not sure how big Wanalancet is, but you go through Wanalancet, but I think it's just Tamworth. This guy's in my backyard as well. So they're around. This is your moss challenge. I chose this one, Neckera pinnata. Now this is an old growth indicator. It might just be a big tree indicator. I'm not sure. Forest continuity indicator. It's also called the shingle lichen. It tends to come off the trees in these shelves. And if you look at it really close up, there's really nothing else that looks like this. It's super shiny. And it's also got these waves. The leaves are like wavy. And of course, that has to do with water holding again. Every single little detail out there that you'll see in these guys has to do with how they hold the water, how they control how fast they dry out. It's really important to them. And on this guy, the sporophytes are pretty hidden, but this right here is one of the sporophytes. This is actually a really tropical genus. This is one of the only species that's in our flora. Most of this family is in the tropics. Your liverwort challenge. You knew it was coming. You're like, oh my God, the liverworts. Nobody thinks they can learn the liverworts. You can learn this one though. It's huge. I mean, it really is. So this is an old kind of diseased beech tree in my backyard. And you can see how kind of similar to Neckera, it does come off in these kind of, it points out from the tree quite a bit. It's big. It's big for a liverwort. And the thing about liverworts is they're kind of flat, and their backside here looks really different than their belly side. This is the belly side. This is the back side. So if you look at them on the tree and you flip them, you should see that it looks very different on the belly. And on the belly, it has leaves along the stem here. And I guess the thing I would say about this guy is it's very pinnate. You know that term from ferns, probably. That means that the branches come out pretty evenly on either side. And the leaves are very scale-like. They look like, it kind of looks kind of reptilian, really. So that, and this is also an old growth, it's supposed to be an old growth indicator. But again, it might just indicate an old tree or the continuity of the forest, but it's good to note where it occurs. And so I'm just going to end with some resources for your searches that I think are worth your consideration. This wonderful book by my friends, the Hines. They live in Maine. Met them when I was working in Acadia. It's just a fabulous book. They write the best keys, very accessible keys, wonderful photographs. Just highly recommend this book. It is a book, though. It's not an online resource, but I would still consider it if you're into lichens, if you want to be into lichens. Whoops. And then this resource here is a newer one, Mosses of the Northern Forest. Now, Jerry was just kidding. This isn't just mosses. He did include liverworts. These are also two friends of mine, but Jerry has just done, he's a perfectionist and he's outdone himself with the photography in here. It's free for download. It's a digital resource and they've made it so you can just download the whole darn thing. And that's where I got these beautiful up close photos are from that resource. This one. I can't remember which one of these is mine. One of them's theirs and one of them's mine, but the close-ups are definitely all theirs. And that's a free plea. I mean, you gotta look this up. This is just even worth just looking at just for the beauty, just for the artistic beauty of it. And then if you do accept my challenge, well, no, it's not my challenge. No. If you accept the challenge of the epiphytes, please, a good way to do it is to record your locations and your photos with the iNaturalist. It's a really great program that you can share data across the globe. But it would be great to see these species documented in our area. I'd be super happy. That's all I've got. Happy to take questions. I know the applause is just deafening. These online talks, I'm just like, okay, hello.
Nat, do you want to stop screen sharing and then we can
Sure.
see faces of folks?
I just wanted to give people time if they were copying anything down.
Are you able to see the chat, Nat, or do you want me to read to you what's in there?
Alice is the body. Somebody answered that. Yeah, so yeah, I meant to mention. So my first photo was epiphytes in the Pacific Northwest obviously are way more obvious than our epiphytes here. And the reason for that is because there's kind of nonstop rainfall and fog. So yes, weather affects epiphytes. The presence of a wetland, like I have in my backyard, will impact the abundance of epiphytes as well. And high on a mountain where the cloud base is, and so cloud base is fog, clouds are fog, the same thing. There'll be more epiphytes there as well. Acid rain, yes, that doesn't sound like a question.
So what's the name of the iNaturalist project?
I didn't make a project, I could. But if you just report them to iNaturalist, at least they'll be reported. So it'll be documented where these things are in New Hampshire. There's very little documentation for epiphytes at all. And
then Kay was wondering, how long do they live?
I would say nobody really has an answer to that. They live as long as the tree lives, and then they die. Once they're on the ground, they'll die.
Guinevere is wondering, do crusts grow on rocks and are they red?
Crusts and macrolichens and mosses and liverworts can all grow on rocks. And which part of them is red? None of them has a really red thallus, but a lot of the cladonias, for instance, will have red reproductive parts, the pycnidia. I guess I'm not sure how to answer that without more detail of.
And then Olivia's wondering
so they're alive. They're dormant. It's kind of like, how does an animal stay alive when it's hibernating? Similar. They're just shut down, waiting for the right conditions to come back. Do larger animals eat like? Oh yeah. So well, obviously not here, but like caribous. Lichen is a huge part of caribou diets. Some people, there's some documentation that white-tailed deer might eat some epiphytes during the winter, sort of starvation food. Pikas in the West also eat lichens. Here, I think I've mostly seen it not used as food, used as nesting material. Squirrels, birds use it as nesting material. I'm getting dizzy. It's moving. Okay. How many species have we lost to air pollution in the Northeast? A lot. If you look at the Heinz's book, they have a wonderful several chapters of background, and one of those chapters is all about looking at historical records and how many of the species are no longer found here. And that's how we know that they used to be here, because they were collected here historically and put in herbaria or photographed. Not photographed. Sorry. Now you can photograph them. Back then, put in herbaria anyway. I'd have to go get my macrolichens book to answer that question well, but my hazard a guess, I would say about 100. I mean, there's so few cyanolichens left. It's really just like 2, 3 with any consistency in most of the Northeast, except for coastal Maine. All right, somebody's gonna go out with their spray bottle. Awesome. Spores that looked red. Yeah, they can look red. They can look brown. They can look green. Medicinal properties. The lichens, likely. A number of the lichens are edible, although I'm not going to go there because they have enough problems. I don't think the mosses and liverworts don't have medicinal properties that I know of, although there's a lot of anti-cancer research, which is looking into any sort of unique chemical chemistry that could be used as anti-cancer, and there were some mosses in that list. So I guess there's no indigenous medicinal uses, if that's what you're asking. There was indigenous use of mosses for diapers and for water absorption because they're super good at that. And they also tend to be antiseptic. Some of them are acid enough, like the sphagnum mosses, which aren't epiphytes, but they're acid enough that they were also used for medical pads during World War I when there was a shortage. Have more. Yeah, that's sphagnum. Cool. Glad some people enjoyed it. Thanks for telling me you enjoyed it. It's hard to judge when you're alone in your bedroom. Cool.
Thank you.
Great. Well, Nat, thank you so much for joining us for our presentation this evening. And we will post a recording of the video on the Squam Lakes Natural Science Center's YouTube channel so folks can, if they want to rewatch it or tell their friends, they can go there to do that. So thanks everyone for joining us tonight, and we hope to see you next month.
Thank you.
Cool.
Thank you.
Good night, everybody. I'm losing my voice.